I would like to welcome everyone to today's seminar, which is on the title of Homeostasis Drives Intense Trace Metal Trafficking on Marine Particles. So my name is Pavla Debeljak, and I started my postdoc in the Blue Cloud project last week with Lucy Bittner. I would like to present myself shortly. So I worked in Bagnols-sur-Mer the last 3 years on my PhD thesis, which was on Southern Ocean agency areas and the microbial cycling of iron and carbon. And prior to this, I was working in a Dutch startup on microplastics and microorganisms on microplastics. Before that, I was in Guadeloupe by a Leonardo da Vinci Fellowship, which is a European exchange program. And I was there working on sulfur bacteria on mangrove roots. And I finished my master's studies in ecology at the University of Vienna, which was already on the temperature dependence of microbial communities in the deep Atlantic. So I would say I'm interested in any sort of microbial processes in a diverse set of marine environments. So to introduce you to today's seminar, I'd like to just shortly introduce you to the Southern Ocean, iron and phytoplankton. So on this slide, we see two maps. The first is the sea surface nitrate concentration, and the lower one is the sea surface chlorophyll concentration. Oceanographers have been interested in the Southern Ocean since the 1930s, and they postulated the so-called Antarctic paradox. So you see in the upper panel that there's a high concentration of nitrate in the Southern Ocean compared to quite low concentration of sea surface chlorophyll. In 1990, then John Martin postulated the iron hypothesis, basically introducing the concept that the lack of iron in these areas causes this discrepancy. And there have been several artificial and natural fertilization studies with iron, and that they demonstrated that phytoplankton is indeed limited by iron. And these areas are called the high nutrient, low chlorophyll or HNOC areas. Additionally to iron, the Southern Ocean shows the lowest concentration of dissolved organic carbon, so of about 50 micromolar in surface waters. This is primarily due to the low productivity regimes, which, as I said before, are because of the lack of iron. And second of all, there's also the upwelling of deep waters, which is mainly of refractory nature. So taken together, this is a picture I took on one cruise I went on. Iron and organic carbon limit heterotrophic bacteria in the Southern Ocean. And why is this a problem for prokaryotic cells? So if we look at iron and carbon in the cell, dissolved organic carbon is the major source of carbon and energy for heterotrophs. And when it comes to iron, bacterial cells out of culture, we know, have a high cellular iron quota, so the amount of iron per carbon is elevated. Furthermore, in the membrane, we have the respiratory chain. Here you have the different complexes of the respiratory chain and the amounts of iron that are in each of these enzymes. So you can see that there's quite a large amount of iron needed for the respiratory chain enzymes. And it's actually 90% of the cellular iron is in the respiratory chain. Last but not least, iron is needed for cellular carbon metabolism. So for instance, the Krebs cycle, the TCA cycle, and several of these enzymes need a cofactor in iron. Right. What are the different forms of iron in the ocean? So very well defined is the dissolved iron, lower of 0.2 micrometers. And then we have these three big pools. One is the iron ligand bound, so the complexed one, the iron 2 and the iron 3. And then there's the particulate iron pool, which is above 0.2 micrometers. So in this study that I'm going to present today, we were actually focused on this particulate iron pool and the question of how bioavailable this actually is. So to look at this, we had basically two questions. First of all, are marine particles a source of available trace metals for heterotrophic prokaryotes? And furthermore, are prokaryotes attached to particles able to use these trace metals that are on these particles for their metabolic purposes? The sampling site in this case was the Kerguelen Plateau. So you see here the map of the Southern Ocean and the Kerguelen Plateau on the right side. This is the Kerguelen Island, and the samples were collected actually close to Heard Island, which is also still on the Kerguelen Plateau. On the yellow point, we have the sample collection of the particles, and the blue point is the sample collection for the waters, so the agenal seawaters, which were the high nutrient, low chlorophyll waters. To a little insight, so this is Heard Island. Actually, on this cruise, it was also observed that there was a volcano, so the Big Ben volcano on Heard Island, which is actually an active erupting volcano in Antarctic waters or in sub-Antarctic waters. So this picture was taken during this cruise. So we collected these samples using in situ pumps. So they were collected at 100-meter depth. So this is the filter, which you see in this picture with this particulate matter. This filter was then resuspended in the trace metal clean 0.2 micrometer filtered seawater from the agenal sea site. And it was added to inoculums of bacterial community water from the agenal sea site as well. So as a conceptual framework, we have the particle and the seawater surrounding the particle, and we have the cells that are attached to the particle and the cells that are in absence of particles or in the surrounding of the particle. So that was the idea that we were going for with this experiment. In order to study the bioavailability or the availability of trace metals, we use 3 different approaches. So there's the geochemical analysis, so splitting the trace metal content of the particles in different pools by chemical leachate. There's also looking at prokaryotic activity to infer bioavailability through production of bacteria. And there's also the gene expression response, so targeting metabolic functioning. So I will first show you the results of the geochemical approach. So this is a plot that actually shows the log of the ratio of the individual trace metal by phosphorus in the cell. So here you on the x-axis you have the different trace metals that we looked at in this study. I will mostly focus on iron, but what we see for iron actually holds true for all the other metals. And when we first look at these lower box plots, these are reference values from culture studies, so from known heterotrophic prokaryotes. So their ratio of the specific metal to phosphorus in the cell, and then in the above blue boxes, we see the box plots that are for our particles. So this is the ratio that was calculated for the particles that we collected and for the specific metal to phosphorus ratio in this particle. And the first result that we see here, so that the ratio of these 7 metals, where we also have the references for heterotrophic prokaryotes, is that the particulate matter is a potential source of trace metal, given the fact that it's higher in these particles than what we know for culture studies. So second of all, we observed prokaryotic activity throughout the experiment on board. So it's basically to infer the bioavailability for the production. So if something's happening, if the cells are growing, or if there's production of biological matter. We observed a 2-phase response of the prokaryotic community. So in this plot, you can see the 12-day experiment on the x-axis, and on the y-axis, you have the cell-specific prokaryotic production. So it's really per cell. And we saw these 2 phases where in phase 1, we have a transient microbial response to the leaching of the product. So the first 4 days where these particles were incubated and the leaching of the organic matter out of these particles, which was fast, which was used fast, or it was, it was recycled fast. And in the second phase, we see a quasi steady state, so where the cell specific production exceeded the control in this last point 15-fold. And this is where we took the samples for our metatranscriptomic study. So this second observation indicates also that these particle attached cells were highly active throughout the incubation period. And in the last part, which is the most revealing or biologically revealing part, is where we looked at the gene expression response. So we targeted the metabolic functioning. Just to have like a brief overview of the bioinformatics pipeline that we used. We had triplicates of the particle attached samples and the control samples. It was, so we did RNA sequencing, so the metatranscriptomic sequencing, also called for the whole microbial community. And this was cDNA depleted, so it was really only messenger RNA, and the ribosomal RNA, which was still left, was then removed bioinformatically. We did an assembly approach, and we mapped back the reads to this metatranscriptomic assembly. We used BLAST-P for the prokaryotic annotations and eggnog for the functional annotation, and I will further focus then on the KEGG annotations, so really the functionals, the protein annotations. And I use differential expression analysis for these samples. So for the differential expression analysis, just to go back to the conceptual framework a little bit, there was this particle that was incubated in the with the agenal seawater. So we have the particle attached and the cells in absence of the particles. So I calculated differential expression analysis one against the other. So when we say looking at metabolic functioning, in the study of trace metals, or especially iron, one very specific concept is, or one very specific mechanism is the transport by siderophores. So these are these ligand-bound iron parts. So this is a siderophore as an example, so a chemical structure that can capture iron. There are 3 different types. I'm not going to go too much into detail, but in the marine environment, they're the amphibic siderophores. And how does this work? So a bacterial cell basically releases a siderophore. This siderophore attaches to the iron-3 in this example, and the bacterial cell captures this by a very specific TON-B transporter system. So it's a bit as a key and lock principle. And so further in this analysis, after I got the results from differential expression, I really focused on the siderophores, so the uptake of siderophores, so specific for iron, the iron-2, the iron-3, and different other trace metals that I've mentioned before, so zinc or manganese or nickel, copper, and cobalt. Furthermore, I also targeted this synthesis of siderophores, because so it's this very costly procedure of producing siderophores for a cell. So it's also very interesting to look at who is or how are they investing into the production of these siderophores. So when we look at these results, so this is a log 2-fold change plot where you see the log 2-fold change on the x-axis, and on the y-axis you have the different proteins that I identified using eggnog, so the KEGG proteins. And these are responsible ABC transporters, siderophore biosynthesis, and the outer membrane receptors for the siderophore biosynthesis. What we see here is that for the cells that were not incubated with the particles, we have a high expression or a differential expression in all of these, for all of these proteins. And when we look at the ones that were attached to the particles and not attached, so if we compare these two, we see that for several ABC transporters, there's high expression, so especially for the iron ones, but for the outer membrane receptor as well, and a high expression of the siderophore biosynthesis genes. So in both cases. So when we take this together and we look at one specific one. So we open up this box of one protein, and we can, because it's a metatranscriptomic approach, we can look at the iron-specific ABC transport, for instance. So for iron 3 or for iron 2. We see that there is quite a high diversity of different bacterial orders, in this case, that are responsible for this. So we see, for instance, Rhodobacterales or Rhodospirillales, which are both alpha protobacteria, being mainly responsible for the uptake of iron 3, while we see Selvipionales or Flavobacteriacea, which are both out of the FCB cluster, so the Flavobacteriacea cluster, responsible for iron 2 transport. When we go for another one, which is, for instance, the siderophore biosynthesis and acquisition, so these called catecholat synthase, which have actually not been observed before in the marine environment, we see that there's a very distinct group, or quite a high distinct group of different species that are expressing this synthase, so the production of the siderophore. But we only, in our case, in this fingerprint of the metatranscriptome, only see 2 SAR groups that are expressing the receptor. So this is quite an interesting finding. So taken together for these uptake mechanisms and the siderophore biosynthesis, we see that there is strong investment into siderophore biosynthesis, which basically tells us that there's trace metals, or there's iron that can be processed through siderophores for the bacterial cells. There's also the investment for the outer membrane reporters and the iron 2 specific transporters, which is higher in the controls, and the control where there are no particles, so the lack of it. And we also see that there's this distinct group of bacteria responsible for these processes. So it's a very diverse process. So in the second part, I looked at the intracellular regulation of metals. So first we had these transporters, but what we were going for mostly was, how is this actually regulated in the cell or in the different bacterial cells? So there's one specific uptake regulator, which is called the FUR, the ferric uptake regulator, but this is a specific case for iron, but it holds true also for nickel or for zinc. It's the NUR or the ZUR, which is responsible for the inner regulation of trace metal, which is coupled with the homeostasis, so iron homeostasis or metal homeostasis of the cell, which is further coupled to cell signaling. So whether a cell is deprived of the metal or has too much of the metal. And why is this important? So I'm just going to briefly explain this because I think it's a little bit complex maybe. There's transcriptional regulation through a 2-component system. So you have an outer membrane, the inner membrane, and you have this outer receptor, which is a cell signaling mechanism for iron in this case. It then gets transferred through the outer membrane, is in the periplasmatic space, and is then transported into the inner membrane. And this all works through an ATP pump. And once the iron is in the cell, it's depending on whether the cell is in iron starvation or in iron surplus, it goes into siderophore biosynthesis or not. And this is all regulated by homeostasis. So homeostasis is the mechanism that actually controls this. And this is by these intracellular regulators. And why is this important? This is important because if there is too much of one trace metal, so whether iron or another metal in the cell, it can become toxic, and this is why the cell needs to eject it as well. So in our case, it's kind of interesting to see whether this was upregulated, downregulated, or whether this stayed stable for the particles. So when we look at again our conceptual model, we see that a high up to 5-fold increase or difference in the expression of the cells that are in the water surrounding and the ones that are attached to it. But even in the ones that are attached to it, when we look at the homeostasis genes, they were all highly upregulated, so highly expressed. So homeostasis was really the driving, or in this concept, the homeostasis for the specific metal, in this case iron, was driving the processes that cause the transport through the membrane of the metals. So taken together, when we look at this trace metal uptake and the siderophore biosynthesis, the expression of these 4 genes is higher in the particle attached compared to the control. We see the differential expressed homeostasis. So there is, if there is enough iron in a cell, the cell needs to regulate it. Bacteria with and without particles differentially express transcriptional regulators. So this is also a very new finding for these sorts of environmental analysis. Taken together, we see that particles are a source of available iron in this case, when we look just at the 4, and that a part of the transport is mediated by this catecholate-like siderophores, which have not been observed before in the marine environment. We see that there are distinct groups of prokaryotes that are responsible for siderophore synthesis, as well as for the uptake. And we see that particulate source of metals requires intracellular regulation in prokaryotes. So this source of metals requires homeostasis to regulate its inner processing and the transport of metals. So with these results, we actually took quite a known concept, which was called the microbial Ferris wheel, which was introduced by David Kirchmann in 1996. And we have here a schematic overview of the microbial Ferris wheel that is called, which is driving the pelagic iron cycling. So you here have the iron pool of inorganic, organic, or colloidal iron. And then we know we have siderophore mediated uptake, colloidal uptake, and dissolved iron uptake. So after the initial uptake, the iron is recycled by heterotrophic bacteria, and then further to flagellate, ciliates, mesozooplankton, and also the viral lysis that again releases iron to the marine environment. And if we, so in this concept, this was also always really based on the dissolved forms of iron. So in this case, we add the particulate form of iron, which is also already directly accessible to heterotrophic bacteria, as we can show with our experiment. So the direct, the uptake and the ultimate release of iron from heterotrophic bacteria may greatly affect iron availability and primary production. So in this sense, with our experiment, we propose to enlarge this idea of the microbial Ferris wheel to the microbial metal wheel. And with this, I would like to thank you for your attention. I would like to thank, of course, all my collaborators and co-authors. We submitted this a few weeks ago. And also ABIMS, because I was working a lot on the Roscoff platform. And if you have any questions or like to discuss and we cannot do it in person, you can send me an email. Thank you very much.